Welcome to the two final lectures in this course section on ecosystem vitality. In these lectures, we will discuss the biodiversity of freshwater ecosystems. My name is Ian Harrison, and I'm a freshwater specialist in the Moore Centre for Science at Conservation International. Biodiversity is the term we use to describe the variety of life on Earth, from genetic diversity to species diversity, community diversity, and ecosystem diversity. It includes the ecological and evolutionary processes that sustain it. It's an enormous topic, even when restricted to freshwater systems, which is why we've split this into two lectures. We'll focus mostly on the species and their importance for ensuring ecosystem health. For a broader discussion of biodiversity, you could read the online chapter on What is Biodiversity in the book Life on Earth by Niles Eldridge, or the teaching module by Harrison et al. That is associated with it. The links to these are included in the resources. In this first lecture, we'll start off by giving an overview of the richness of the freshwater biodiversity itself. We'll describe the taxonomic diversity of species and the geographic diversity in terms of where they're found. And then we'll take a quick look at how this biodiversity supports a rich web of ecological interactions and how this ecological functionality of freshwater ecosystems is important, not only for the ecosystems themselves, but also for the people who rely on the natural benefits, that is, the ecosystem services, supplied by healthy freshwater ecosystems. Then we'll look at the human effect on this biodiversity, how we're causing its decline, and what we need to do to rectify that. And then we'll look at how we obtain data. We'll briefly discuss obtaining novel field data and how to ensure that we do this monitoring in a standardized way. In the hands-on activities associated with this pair of lectures, you'll be able to take more of a look at finding biodiversity data and applying it to the Freshwater Biodiversity Indicator of the Freshwater Health Index, as well as other indexes. And you can look at some examples of recent scientific papers that discuss ways forward for better integration of freshwater biodiversity information into research, management, and policy, and decide what recommendations seem most important to you. The Freshwater Animal Diversity Assessment estimated the number of scientifically described freshwater animal species and vascular plant species to be about 126,000. This represents about 7 to 12% of the total number of scientifically described species on Earth. However, the Freshwater Animal Diversity Assessment is old. It was published in 2008. A more recent source, the website of the International Union for the Conservation of Nature, usually referred to as the IUCN, estimates there are over 140,000 described species that rely on freshwater habitats for their survival. By this we mean species that accomplish all or part of their life cycle in or closely associated with water, like this fish, for example, or species that show a close dependency on aquatic habitats for food, for example, like this fishing cat that feeds mainly on fishes. 12% of all described species might not sound too much, but note that freshwater covers only about 0.8% of the Earth's surface, so, relative to area, that does represent an amazing richness. And one important thing to note for the species numbers provided by the Freshwater Animal Diversity Assessment and for the IUCN Red List is that they're not comprehensive. For example, they tend to cover these groups quite well, but their coverage of these extra groups on the top line is not as extensive, and especially groups like bacteria, protozoans, fungi, and algae, which may have many thousands of freshwater species. So this number of 140,000 species is certainly an underestimate. Also, notice that freshwater species are well distributed through multiple taxonomic groups, from simple bacteria and eukaryotes or protozoans, through algae, plants, many groups of invertebrates, and up through vertebrates. And some of these groups are exceptionally rich in freshwater species. For example, some families of crabs are only found in fresh water. Over 60% of amphibians are found in freshwater, and 50% of fishes are freshwater species. The Freshwater Animal Diversity Assessment also looked at the biogeography of freshwater species. Where are they most abundant? The mapping is rather coarse because they're mapped to very large geographic regions, so we can only make general comments about these freshwater distributions. The large Palearctic region, marked PA, is the richest in freshwater species. Then you have the continental tropical regions like the Afrotropical region, marked as AT, the Oriental, OL, and the Neotropical, NT regions. Now, usually species richness is greater at the tropics, so it's surprising that the largely temperate Palearctic region has more species than these tropical zones. But that is in fact an artifact of sampling. The Palearctic is well surveyed relative to other areas, and especially for species-rich groups like insects. Stoneflies, caddisflies, and various dipteran flies show high levels of species richness here. Crustaceans and mollusks are also quite prevalent here. If we look at the tropics, the neotropics has large numbers of insects, but it's particularly interesting that we see much greater numbers of vertebrates, especially amphibians and fishes. This is true here and in other parts of the tropics. And this is of course because of the huge area and diversity of freshwater ecosystems found in these tropical areas. For example, the Amazon, which you see on the top right, is the greatest interconnected fluvial system in the world. The Pantanal in southwestern Brazil, Bolivia and Paraguay in the photo below is the world's largest wetland at 210,000 square kilometers. There are of course other data sets that show global distributions for freshwater biodiversity, but they tend to be focused on specific taxonomic groups. For example, this data set for fishes. These can show us particular basins that are very rich in fish species. For example, I've worked on the Congo Basin, which is rich in fish species with more than 1,200 described species, and about 75% of the riverine species are endemic. I'm particularly interested in the lower Congo section, that is the 350 kilometer long stretch running from Malebo Pool by Kinshasa down to the sea. That small stretch of river has a very unusual hydrology with complex flows and multiple large rapids. You see the Inga Rapids here. You'll see another view of it later, highlighting some threats. Species diversity and adaptation have been driven by these conditions. There are about 446 species of freshwater fishes in this section of the river, and 30% are found nowhere else. That's more than all the species of freshwater fishes in all of Australia. And all of Europe has only about 1/5 more species than this short section of the Congo River. We'll come back to this example later when we discuss the Freshwater Health Index biodiversity indicator, but I mention this as just one example. We could pick any other freshwater group and find similar examples of extreme richness at particular sites. So, the living organisms and their interactions among themselves and with the physical environment are what makes freshwater ecosystems dynamic, self-sustaining systems. And this helps deliver an enormous range of ecosystem services, from fish and other species for food and medicines, through to regulating processes of water filtration, nutrient cycling, and of course to aesthetic and recreational services like walking, diving, bird watching, dragonfly watching, or angling. Now let's look at some of the threats to freshwater biodiversity and what we need to do to address them. In 2006, David Dudgeon and multiple co-authors reviewed the threats to freshwater biodiversity, and in 2019, Andrea Reed and her co-authors published an update to this, looking also at emerging threats. A link to the open access paper by Reed et al. Is in the resources. So this slide shows the general threats that Dudgeon et al. Identified, and I've added climate change onto that. These threats are closely linked, so that something like habitat degradation can alter flow regulation and vice versa, and both components threaten biodiversity. Current assessments show that the threats to freshwater biodiversity are very high. For example, the Freshwater Living Planet Index compiled by WWF shows that assessed species, which are albeit only for a restricted group of vertebrate taxa, have declined since 1970 by 84% on average, as you can see from the graph on the right. There are not only declines in populations, there are also extinctions. For example, most evidence now points at the extinction of the Chinese paddlefish, which you see in the photo on the lower right. An example of charismatic megafauna of freshwater systems. So we have a list of about 80 species of freshwater fishes declared extinct, according to WWF. In 2020, a group of scientists suggested how we might address this pattern of loss in freshwater biodiversity, and this included 6 main themes of action: improving flows and connectivity, water quality and habitat, and exerting better control on exploitation of species and invasive species. Thinking about how we assess the health of freshwater ecosystems, some taxonomic groups are especially sensitive to the state of the ecosystem and are very good indicators. For example, mollusks are sensitive to water quality, some fishes are very sensitive to water turbidity, dragonflies are an excellent group because their life history is dependent on the freshwater ecosystems for the larval and adult stages, but also on the riparian vegetation adjacent to the rivers and streams for the adults. One of our course co-lecturers, Dr. John Samaika, has assisted in the development of a dragonfly biotic index using the presence of dragonfly assemblages to assess the status of the freshwater ecosystems where they're found. So let's think more about where we get our biodiversity data from. In some cases, this might require going out into the field and doing monitoring. In those cases, it's important that the surveying, sampling and monitoring are done in a standardized, accurate and consistent way so that they can be replicated at other locations and times and the results are comparable. There are numerous sources online for defining sampling protocols, for example, surveying for freshwater invertebrates or for fishes, and the links to all the documents I'll show here are in the resources, so don't worry about trying to note them down. And if you do an internet search, you'll find many more. There are more general guides, such as Conservation International's Handbook on Methods for Biological Field Assessment, and IUCN's Guide to Good Practice for Wetland Assessment that includes guidelines for biodiversity surveying. This concludes the first part of our lectures on biodiversity. We've looked at how freshwater ecosystems are among the richest on the planet in terms of the diversity of species compared to the available area of freshwater habitats. We've seen how these ecosystems and the species they support provide a wealth of important ecosystem services to people. But they are the most threatened ecosystems on the planet compared to those of marine or terrestrial biomes. And we've seen how assessing the status of freshwater biodiversity requires assimilation and analysis of species data, which requires standardized and reliable in-field techniques of biodiversity monitoring. Thank you for your attention. Please rewind and replay any parts of this lecture that were not clear to you. You'll also find a full set of resources cited in this lecture on the course website. There are 2 voluntary hands-on activities associated with this lecture. The main hands-on activities will be presented in the second lecture.